Hi, my name is Meagan Olsen, and I am an undergraduate student in the lab of Dr. Shannon Servoss at the University of Arkansas. My research is centered around the functionalization of spherical gold nanoparticles with peptoids that can bind to viral envelope proteins. Peptoids are poly-N-substituted glycines that basically mimic peptides, but their structural changes mean that they have improved protease resistance, which is important for biological samples. The idea is that by coating gold nanoparticles with peptoids, we can develop electrochemical sensors with improved response times, specificity, and accuracy. We have focused on a proof of concept peptoid capable of binding to green fluorescent protein, or GFP. Nanoparticle functionalization is complicated by the fact that adding peptoid to a solution with nanoparticles leads to the nanoparticles aggregating. This aggregation is accompanied by a shift in color, can be monitored using UV-vis spectroscopy with the shift in the peak location, and is increased by an acidic pH or sample mixing. Addition of phosphate buffered saline also leads to rapid aggregation because the salts present in that solution disrupt the stabilizing citrate layer on the gold nanoparticles. To prevent aggregation during functionalization, an intermediate monolayer of tween 20 can be used to sterically stabilize the nanoparticles, making up for the displacement of the citrate layer and its electrostatic stabilization. This allows for nanoparticle functionalization and isolation without losing any of the nanoparticle functionality. To confirm that the peptoid actually bound to the gold nanoparticles and retained its ability to bind to a protein, the functionalized nanoparticles were incubated with GFP and then isolated. The functionalized gold nanoparticles do demonstrate an appreciable fluorescence after centrifugation and resuspension, which indicates that the peptoid was not only able to bind to the gold nanoparticles, but it retained its ability to bind to GFP. That being said, there is variation between the representative trials shown here, and further work is needed to clarify these results and relationships. Nanoparticle size also has an impact on the functionalization process. 10 nanometer and 40 nanometer spherical gold nanoparticles were prepared and given similar solutions to be tested with, and they gave similar functionalization results. However, the 40 nanometer particles were less likely to adhere to the plastic centrifuge tubes and were not as impacted by the addition of methanol, which was found to promote aggregation of the functionalized nanoparticles. Even in the unfunctionalized nanoparticles, addition of methanol causes a slight peak shift in the 10 nanometer particles, while addition of water to the 40 nanometer samples actually decreased in intensity and caused the 40 nanometer samples to be slightly less stable. Over the course of this research, we determined a method to coat gold nanoparticles with peptoid without losing peptoid functionality, and noted that there are some size-dependent properties that need to be further explored. I'm working to extend this method of functionalization to another peptoid of interest designed to bind to a viral envelope protein. We can then incorporate the nanoparticles and peptoids into an electrochemical sensor, hopefully improving point-of-care viral diagnostics through our simple, rapid method.